restrictions have been busy on a number of fronts. I'm just going to share a few things with you. Probably one of our most visible efforts over the last 3 or 4 months has been our stewardship activities. Uh, and I just wanted to say that our next scheduled stewardship day is this coming Sunday, March 14th from 9 to 12. And you can learn more about where it's going to be located. It's going to be up in Alden on the Keebler parcel. But all you have to do is Google Friends of Hackmatack National Wildlife Refuge, and you can get more information about how you can register and participate in that effort. Um, Also, I'm delighted to share with everyone that our next scheduled Friends gathering is Tuesday, uh, June 8th. This event will be hosted by the Village of Spring Grove, and we'll have an opportunity to tour the revitalized Spring Grove Fish Hatchery. Uh, and again, Google Friends of Hackmatack, and there'll be more information as that event nears. And then I want to just update everyone that and thank the volunteers and everyone that has been participating in our Illinois Clean Energy Stewardship Grant. We're very close to meeting all of the challenges. And that will result in approximately $34,000 being used for seed to restore prairie and oak savanna at a location just north of the Richmond Burton High School. And I really need to acknowledge Cindy for getting the word out, for Pete Jackson for coordinating the events. Again, the volunteers have been a huge part of this. And then also the students with the Richmond Burton High School have been participants, and I'd like to acknowledge and thank them as well for their participation. Um, so finally, on behalf of the Friends, I'd like to take this opportunity to wish you well, be safe, and enjoy the upcoming spring season. And with that, I'll turn it over to Todd Boonstra, our refuge manager.
Thanks, Steve. I'll keep this short and sweet because obviously I know that nobody came on here just to hear me talk. COVID I figured I might as well circle back with COVID, since obviously COVID has been the center of everybody's life for the last couple of years. And I just last year. And I just wanted to reiterate with everybody that if you're anything like me, getting outside has been a lifesaver this last year. And when it comes to 2020, 2021, we're actually really excited to still have that opportunity. The refuge is open for everybody to come out, visit, take a look. There is a slight change for this year in regards to on federal properties. There is a mask requirement for this year. And I just figured I better bring that up. So I strongly encourage continue to wear masks. If anything, make sure you got social distance when you come out to visit the refuge properties, but they are still open. Go out and enjoy them. I was just out there today. It was a gorgeous day to be out there, and it was nice to see the snow finally disappearing. So I'll keep it to that. If you have any questions, by all means, give me a call, and I'll throw it back to Greg to introduce Kathleen.
Well, thanks very much, Todd. I appreciate it. And without further ado, I would like to introduce our keynote speaker. Kathleen Marie Garnes has been fascinated by orchids for many years. Her artwork was featured by the Smithsonian Institution at their 2010 exhibit, Losing Paradise, Endangered Plants Here and Around the World, and on the Smithsonian's North American Native Orchid Conservation website. She also recently illustrated the new glossary for Flora of the Chicago region and 2 guides for the Field Museum. And right now, Kathy is working on a guide to the ferns and an oak woodland habitat guide. She is the scientific affiliate of the Field Museum of Natural History and research affiliate at the Morton Arboretum. Take it away, Kathy.
Unmute. Can you hear me?
There we go.
Okay, let me see if I can pull this window up. Can everybody see that?
Not yet.
Okay, hang on. Share screen. Now, can you see it?
Yes.
All right. Well, I was hoping to get a full screen here, but it doesn't seem like that wants to do it. But so rather than just bear with me, the images will be a little smaller than I'd like, but the content will be the same. A lot of people aren't aware that there are over 50 species of orchids in Illinois and probably a comparable amount in Wisconsin. Of those, 42 species of native orchids were part of the Chicago wilderness pre-settlement landscape. According to, and I'm using the flora of the Chicago region, plants of the Chicago region as my authority here. So if I refer to Wilhelm, we know that it's Dr. Gerald Wilhelm from Conservation Research Institute. I'm also just in the housekeeping, when I mention the orchids, assume that it's a C value of 10. I hope everybody knows what a C value is. It's a coefficient of conservatism, which indicates a percentage of confidence that what the plants that you're seeing there are in a remnant habitat. So a C of 10 is a 90 to 100% degree of certainty that this plant that you're seeing is you're standing in a remnant habitat, and a 0, for example, you would have no degree of certainty that you were in a remnant habitat. So I just want to kind of get that out of the way, because some people know this and maybe others do not. But assume that when I talk about these plants, that unless I say otherwise, that they're a C of 10. Most of our orchids here are very rare. In fact, 25% of those 42 have been deemed extirpated from Illinois. And another 25% are listed as state or federally threatened or endangered. And the rest of them are under serious threat. And we'll talk about why in a minute. A lot of people aren't even aware that vanilla is an orchid. So here's a photograph in the center of a cross section of a vanilla flower, just so people are clear. The one on the left is the large world begonia, and not Begoniophylla ascites, but the other begonia that only lives in southern Illinois. And then we have the yellow lady slipper orchid on the right. So we have both the extraordinarily rare, and you have the what we would like to think of as ubiquitous, but unfortunately they're very rare also. Orchids are very charismatic. So they are sort of the plant family equivalent of the panda. They draw attention to the remnant or aboriginal habitats, and they really, in my case, certainly compelled me to want to save them. They are unique among plant families in the fusion of the male and female reproductive organs into something called a column. They're one of the 2 largest and certainly the most highly specialized and also the most morphologically diverse of all the flowering plants in the world. They are sensitive to subtle changes in the soil environment. They're entirely dependent on mycorrhizal fungi for sprouting and for also bringing them to a great extent a lot of the nutrition. And some of these are more dependent on the fungi than others, but at the very beginning of their life cycle, they are entirely dependent on these mycorrhizal fungi. They Givnish et al. Has identified the antiquity of orchids to go back to about 112 million years. So here's a little gallery of just McHenry and Walworth County species. So pertaining just to the Hackensack area. And we're going to break it up a little bit by habitat. I thought that that would make it a little more interesting and easier to remember some of these beautiful plants. The first one is Cypripede macaulay, the pink lady slipper, or sometimes called the stemless pink lady slipper. Unfortunately, according to Randy Neibauer, it is extirpated in Illinois. If it's not, they probably would like to know. It has been vouchered from both Walworth and McHenry counties, from sphagnum bogs and wet woodlands, with plants like Andromeda, northern pitcher plant, poison sumac, and 2 sundew species. The illustration on the right is one that a scientific illustration that I did a number of years ago. Chiloglossum viridae, and please forgive any misspellings. I know botanical Latin is, it's a dead language and everybody has their own pronunciation of things. It likes bluffs and slopes in mesic woodlands. Wilhelm describes it as being found on humus-covered, decomposed logs in sphagnum tamarack bogs. Its former name was Dactyloryza viridis. The Ojibwe considered this plant a love charm. And it's a spurred orchid with probably one of the widest distributions of all the orchids in our region. That's worldwide. The coral roots are one of those saprophytic orchids that need the fungi exclusively. They are called coral roots because of the structure of their roots. They look like aquatic coral. And they, the one on the right, the autumn coral root, shows its clistogamous form. It very rarely has a chismogamous or open flowered, open pollinating form. And like some violets that also do that. And I think a number of species of plants both have specific flower forms that encourage self-pollination versus open pollination. The coral roots also do this. And it may just be a response to lack of pollinators. We're really not sure. But the coral root, spotted coral root, has been recorded from Walworth County. And they're all the coral roots are saprophytes, meaning, as I described before, they get all of their nutrition from the soil layer. There's very, very little photosynthesis happening here. The word maculate means spotted. Immaculate means unspotted. Those both have a C of 10. And they're really interesting. They look like little brown asparagus when they come up. Here's my yellow lady slipper painting that hung at the Smithsonian, Kew Gardens, London. This is another one of those beautiful, charismatic lady slippers. And it's much more rare now than its distribution suggests in the Florida, Chicago region. It's a favorite of deer, as are most orchids. And the remnant populations are usually only on mesic wooded slopes, kind of well out of harm's way now. The showy orchis likes kind of open mesic woodlands. This is another one I've monitored for plants of concern. It's another species that's been predated by deer, but it's mostly been much reduced in numbers due to canopy closure and lack of fire management. Goodyear pubescens, downy rattlesnake plantain, gets its name from these beautiful little fuzzy flowers and the appearance of the The evergreen, the netted evergreen leaves sort of look like a snake skin. They like wet to dry mesic woodlands, and the rosettes are about 4 inches across. They're very small. They're very delicate, kind of diminutive plants, very easy to overlook. The best time to look for them actually is in the fall or the winter, when you can the evergreen rosettes are much more apparent. A couple of considered weedy orchids. The purple tway blade is probably one of the most adaptable and least conservative of our native orchids. It'll often pop up in wooded areas that were recently restored. This has a C value of 4. And a lesser lady's trusses is considered by Wilhelm to be adventive to our region. The Spiranthes ovalis, variety Aristolata. And its earliest collection in our region was in DuPage County by Wayne Lampa in 1981, but it does seem to be expanding its range. This last year, Paula, Evan, and Kelly Schultz and I discovered a county record of it in Lake County. So we were excited about that, even though the botanists think, oh, well, it's another weedy orchid. I don't care. It's an orchid. I'll take it. Herman Papoon talked about orchids, just being as far as the eyes could see in our prairies. Unfortunately, we have less than 1/100% of orchids of our prairie habitats left. And so all the more reason to take just the best care of them that we can. Here we have two very similar orchids that bloom at different times and that are slightly different. These are fairly good photographs to describe what I want to tell you a little bit about these. The Oklahoma grass pink, you might not know, was actually collected in the 1800s in McHenry County at Ringwood by Vasey. It blooms a little earlier, usually a couple weeks earlier than Calopogon tuberosus, the grass pink orchid. It's distinctively, it's slightly shorter in nature. It prefers much more upland sandy habitats. I have seen it in Kankakee County in company with the grass pink orchids about 100 meters downslope. And the Oklahoma grass pink was in capsule already. It was already in fruit, and the grass pink orchid was just starting to open up. So they live in very different habitats. But I'm kind of curious to know that it had been identified in McHenry County at one point. The one way to tell them apart is the Oklahoma grass pink. The petals like curve upwards a little more. They're usually a little lighter color, but the giveaway is the leaf. So the flowering stem is about the same length as the leaf, whereas with the grass pink orchid, the flowering stem way overtops the leaf. And what's kind of interesting about these two particular species is that they're pollinated by deception. If you see this, let's see if you can see my little pointer here. On both of them, you will see the lips kind of flattened down against the upper petal. What's happened here is they've had an insect attempt to get some nectar from the orchid, and the weight of the insect causes this little spring mechanism to activate, and the lip slams down on top of the insect and traps it, and it has to make its way out and around by the by the column and pick up the sticky pollinia. And hopefully from there, it will go and find another orchid. What I've also found interesting is the grass pink orchids are the same color, exactly the same color as marsh phlox, which do offer nectar. So I wonder how much pollen gets lost to marsh phlox instead of other grass pink orchids. I do not see a lot of, this is one of the plants I monitor, and I do not see a lot of pollination happening. In a population of a couple hundred plants. We'll see, 2 or 3 capsules, maybe, if we're lucky in a particular year. And so that's a real concern. Of course, loss of habitat and changes in habitat are also causing a tremendous decline in these beautiful plants. But usually it's only immature bees. I read somewhere that it's only immature bees, the newly emerged bees, that pollinate these orchids. I guess after getting slapped around a couple of times, they have learned their lesson and they stay away from these, and they go find other less problematic sources of nourishment. But they certainly are a treat when you get to see them in nature. They're really just so beautiful. Oh, one more thing about the calopogons. They respond extraordinarily well to fire management. And I could talk about that more, but not maybe in the chat. So let's go on to Cypripedium andrewsii. The photo here in the center is by Werner Frosch, and I stole it off of the University of Wisconsin Herbarium webpage because it was the most clear example of the hybrid parentage. So you have the dark sepals and petals of the Cypripedium parviflorum macacin parent and the white pouch of the Cypripedium candidum parent, and they just make this beautiful baby together. I have not seen this in person, but it does only happen where you have both parents together in usually moist prairies, fens, and bogs. Wilhelm states that a controlled burning in the early 1990s produced literally hundreds of the hybrid among countless numbers of both parents. Charles Chiviak, who wrote the Introduction to the Ecology of the Illinois Orchidaceae for the Illinois State Museum a number of years ago, reports it from McHenry, but Wilhelm did not find a documentary specimen to support the literature record. Cypripedium candidum lives in calcareous to wet mesic prairies. Again, it used to be ubiquitous here in our region. When I'm monitoring new populations of it, the bright yellow umbels of golden Alexanders kind of give me a hint of where to look. Platanthera flava herbiola is another one that's you seen in wet to wet mesic sand prairies. It's been vouchered from Walworth County. And it's one of these plants, and unless you really know what to look for, it's really good at hiding within the population. But if you have a little hand lens along with you, it's just an exquisite little flower. Going on to some more prairie platantheras, we have the ragged fringed orchid and the eastern prairie fringed orchid. I'm not going to talk really to any extent about eastern prairie because Kathy Pollock and Lori Ryan will treat that in much more detail later. But somebody once told me that these two orchids look alike. I don't think they look anything alike to me at all. I don't understand why anybody could say that. But you will find them in similar habitats. Platanthera lacera was vouchered from Walworth County It has a C value of 7. And the Eastern Prairie Fringed Orchid is always a delight whenever you run into it. In some habitats, it's caused a lot of ecotourism has caused quite a bit of destruction to some of its habitats. So keeping these populations of plants confidential is just so important. It's really a privilege to be able to participate in their life, and we have to be very mindful of the people that we draw into that circle. I hate to say. This is yours truly in what Janine Catchpole calls field position, going after actually Spiranthes incurva. I did not update the title on the slide. Matthew Pace and Kenneth Cameron split it out from Spiranthes cernua sensu stricto in 2017. It's considered really an ancient hybrid between Spiranthes cernua and Spiranthes magnacamporum. It's probably the most frequently found of our native orchids. It has a C of 4. What's distinguished about it is the multiple ranks of flowers and the inflorescence, sort of giving it the look of a lady's braid, especially right before it blooms. The basal leaf blades are usually persistent at the flowering time. And it likes kind of calcareous sand prairies, mesic to dry sand prairies, marley pennays, and even Wilhelm says a high water table to well-drained old fields. A very large patch of it emerged from under the brush in a Cook County Forest Preserve site after 2 years of careful management. This patch was about the size of my bathroom, and it was just so dense. It was ridiculous. At my site, it often pops up in mowed grassy edges, and I have to warn the park district about its bloom cycle. Again, that has a C of 4. Spiranthes magnacamporum looks very similar to Spiranthes cernua, but you have to kind of get down again on your tummy to smell it, or check the leaf blades, which are usually absent at flowering time, but not always. The vanilla fragrance is kind of the giveaway, though, for this Spiranthes magnacamporum. It's also a slightly more robust looking plant to me. But I've seen them. There's a lot of variability, and I see them in the same habitats fairly often. Extra points if you know what this beautiful orchid is. Pagonia ophioglycides. We're going to talk a little bit about orchids of the wetlands. And Thomas Meyer discovered this boreal relic in 1986, and William and Rebecca calls it the swamp pink orchid, which I think is a decidedly unglamorous name. Most of us prefer to call it dragon's mouth. So you can decide for yourself what you think it most looks like. But it's very tiny. This plant's like 5 inches tall, if that. This is an illustration that I did for the cover of Erigenia a number of years ago, and this is a photograph that I took of showy lady slipper in a fen in central Wisconsin. The McHenry County specimen of Cypripedium reginae was from a tamarack swamp where it grew on hummocks. Leporus lacellii prefers fens, marshes, and marley pennays. This the photograph on the left was taken by Linda Curtis, who knelt down, took off her boot, put the boot in behind the orchid to kind of make a point of "Don't tread on me." So you can see for scale what really a very tiny plant it is. But what's also distinctive about it is this almost chartreuse green that's kind of distinctive among other plants. I've only seen it in central Wisconsin in the wooded edge of a fen, and a couple of times along the lake plain. The northern bog orchid, Platanthera aculonis, the Latin name, Flora of the Chicago region considers it synonymous with Platanthera hyperborea and Platanthera huronensis. The flowers sort of to me look like Platanthera dilatata, the bog candles, but there's usually more of those in bog candles. Is a much taller plant. And I've seen bog candles again, not in Illinois, but in central Wisconsin, in the same habitat, actually the same fen as Cypripedium reginae, and a whole bunch of other cool plants. The Aquilonis has been vouchered from both Walworth and McHenry counties. And the plant gets to maybe about 2 feet tall compared to the bog candles, Platanthera dilatata, which can get pretty, like at least 3 feet. The one I saw was close to 3 feet tall. So they're very similar, but very different. Pogonia alphaglossides, a snake mouth orchid, lives in sphagnum bogs, prairie fens. Marley panays, supposedly along Lake Michigan, and sandy marshes. This is another species that I monitored for a while. It seems to me that the numbers drop as the canopy closes. They appear to prefer to me wet edge habitats with some sun. And when the canopy, when you're not managing and you let the glossy buckthorn take over, they just kind of go away. It's one of its BFFs is a northern pitcher plant. And then we have another wetland ladies trusses, Spiranthes romans ophiana. It's only vouchered from 4 counties in the Chicago Wilderness region, but McHenry County is one of those. The earliest record is from 1873 from Glencoe, Illinois, low sandy prairie. But the McHenry County voucher was collected in 1977 from a floating peat mat in a glacial lake. And then we have one adventive non-native orchid, Epipactis haliburine. I think that this one is really, really interesting. I see it pop up in hosta beds for some reason, but there's 3 different color forms. There's a really dark maroon one. Then there's this pink one with a dark maroon center. And then I've seen them also just a clear green and white with no dark center. I have heard that they are invasive in certain habitats. A friend of mine said that she saw it like kind of taking over a wooded area in Vermont. So that is just something to be mindful of. She says when she sees it, she pulls it out. A little bit about orchids in history. I mentioned the fossil record, but they were really recorded a long time ago in the earliest Chinese literature, and it's one of their sacred plants, along with bamboo and plum and another one I can't think of right now. It's been used in European medicine since the time of ancient Greece, and it's had an important role in the doctrine of signatures because of the shape of the roots. It was kind of considered an aphrodisiac. The vanilla first gets mentioned in the 16th century. And then a little later, we have documentation of 15 species of native orchids in Europe. And then Carolus Linnaeus publishes a monograph on orchids in 1740. Lewis and Clark discovers 2 new species of orchids along their travels in 1805. Then you have the famous story of Charles Darwin. 1862, he was sent a specimen of this unknown species. They figured out that it was an orchid from Madagascar, and it had this spur that was 12 inches long. And so Darwin looked at this and he theorized that there had to be a night flying moth, because it was a white flower, that had a proboscis that was 12 inches long. And so he offered that up to his colleagues, and they basically laughed him out of town. What's interesting is 20 years after he died, that moth was discovered in Madagascar, and there are YouTube videos showing the pollination of that moth on the orchid that are up on YouTube right now. Its name was Xanthopan morgani predicta, which is a little nod to Darwin. Darwin knew it all along. Orchids are being studied as indicator species of habitat quality and changes over time. Dr. Melissa McCormick at the Smithsonian has been very active working in this field, and new discoveries of species occur all the time. So what do orchids tell us? We know now that the research on soil fungi indicates that there are many essential interrelationships between species that we don't understand yet. And so because orchids are these charismatic species, people will tend to, throw money at them so that we can learn more. And so everything benefits. We develop new and better scientific protocols to understand what's happening to our surface and groundwater supply and what impacts that will have on humans as well as orchids. The diminishing occurrence of orchids due to fire suppression, shading, canopy closure, hydrologic changes have taught us how really infelicitous our relationship is with the land where they grow has become. Orchids are usually indicative of grade A natural areas, and less than 0.1% of Illinois fits that category, and probably a lot less than that. What are some of the threats to our orchids? We have invasive species, of course, deer browse, fire suppression, hydrologic changes, biocides that kill native pollinators. People are just very free about using pesticides adjacent to our natural areas, and that has had an impact on the pollinators for orchids and other plants as well. Trampling, over enthusiastic photographers and uncareful monitors, poaching, construction and development, and even a general lack of interest in native plants and their habitats, which is a very sad thing, but something that I think we need to continue to fight. Couple of charts that I pulled out for my the presentation that I did for the Native Orchid Conference up in Green Bay a few years ago. Just look at the change from prairie to cropland here. It's heartbreaking. So it gives us even more. I think it should put more of a point of why we do what we do to save what remains. And I'm sorry if this sounds depressing, but this is real. But there are some good news. So you have the prairie white fringed orchid monitoring starting in 1973. We are starting to notice these population changes. Research indicates that invasive species and water quality change are drivers, but there's also good news in that as we do more restoration, new populations of orchids are being found every year in habitat rehabilitation areas. We have volunteers being trained to help restore and monitor these rare plants, and everybody can make a difference. One thing that was very helpful from the Wild Things conference was the land acknowledgement statements that affirm a more congenial relationship between ourselves and the land upon which we and our native orchids live. So why should we care? Well, as I probably hope I've made the point that orchids are indicator species for high quality remnant landscapes whose soils are still intact. With the exception of a very few, the orchids that I mentioned here, the C value 10 orchids, only will thrive and reproduce where the soils are intact and healthy. They're an important part of our heritage of biodiversity on Earth. I'm not going to get theological on you. I certainly could. But, you know, they've been here over 100 million years. And every loss of a native species, of an Aboriginal species, makes us more spiritually depauperate too. They're also one of the largest and most adaptable families of plants on Earth. If they all disappear, well, what does that mean for us? And we have a responsibility. We have a moral responsibility to future generations to preserve maximum biodiversity, to preserve what was created that humans can't make. We can't make an orchid. We can create we can create conditions in a lab that may produce an orchid, but only an orchid can make another orchid. So we have to save them. And how we treat orchids in their habitats reflects how we treat ourselves and the very earth upon which we depend. And then that applies to not just orchids, but every Aboriginal plant, every native plant in our remnants reflect this care or lack of it that we give them. Here's some photos from Workday a number of years ago at Ted Stone of people having a lot of fun, burning up the buckthorn and getting outside and having some good exercise. This beautiful remnant dolomite prairie woodland. He has about over 400 species of native plants there right now. This is the orchid, the prairie. I don't think this is in McHenry or Walworth, but this orchid was my gateway drug into native orchids of Platanthus psychotes. And it still has a very, very compelling presence for me even today. The hands that you see on upper left are Dr. Bill Berger, botany curator emeritus from the Field Museum. He came out to McHenry County to one of their sites and showed us how to hand pollinate the white lady slipper orchid. And here's a little primer on saving orchid habitats. I feel like here I'm kind of preaching to the choir, I think connecting with other advocacy groups and not remaining siloed is what I would focus on. Beyond your field guides and beyond the restoration work that you do, we need to, they say it takes a whole village to raise a child. I think we have to be reminded, not just every other year with the Wild Things Conference, but, continuously to build capacity to find out who does what well, and continue to advocate, continue to educate for the conservation of orchid habitats, and educate your family and your friends too, everybody. So I want to close with, what does the future hold? A little quote from the Lorax: unless someone like you cares a whole awful lot, nothing is going to get better. It's not. And thanks to Greg Reisky and Kettle Moraine Land Trust, and Todd Boonstra and Steve Byers, and Lori Ryan and Ed Collins and Kathy Pollock for all of your wonderful work here, to Suzanne Macy, who raised me up as a Plants of Concern monitor, and now Gretel Kiefer, current director of Plants of Concern, for orchid population data and encouragement, for Illinois Natural History Survey staff, botanist Jeff Hateman for many of these images, the Native Orchid Conference, before whom I first offered an iteration of this presentation. Actually, it was covering all the orchids of Illinois, the University of Wisconsin, Green Bay in 2009. And, all the botanists before us who studied orchids and kept us in the loop about them. I just want to say thank you. And that's all.
Well, thank you very much, Kathleen. I know that if we didn't have everyone on mute, you'd be hearing the applause. So what I'd like to do is move right on ahead with our next follow-on presentation. We'll be able to circle back to Kathy for additional questions after we get a little bit of an update from our next two presenters, who will be talking about the recovery program for the Eastern Prairie Fringed Orchid. So I'd like to introduce our next two speakers. Lori Ryan is the plant ecologist with the McHenry County Conservation District. She's been coordinating volunteers and monitoring rare plants through Plants of Concern program and the U.S. Fish and Wildlife Service Eastern Prairie Fringed Orchid Recovery Program for almost 15 years. And to start us off, Kathy Pollock, who is a biologist with the U.S. Fish and Wildlife Service, who works on the recovery of the Eastern Prairie Fringed Orchid across its range. So, Kathy, would you like to take it away?
Sure. I'm going to share my screen, I think. Can everybody see that?
Mm-hmm.
Okay.
Yep.
So in this initial slide, you can see the Eastern Prairie Fringed Orchid in various life stages, and that's what this plant is, the plant Lori and I are going to be talking about. So this is on the left. Let me see what it looks like. Can you see me pointing when it comes up out of the ground? This is a blooming, reproducing adult, and this is what it looks like with the seed pods later in the season, like in August. And if you look closely at this photo, you can see the seed pods up here are nice and plump on the upper right. And this one is really skinny, and this one is really skinny. It's an indication that these were cross-pollinated and these were not, but I'll talk more about that later. I'm trying to move forward and my enter is not working. Any suggestions with the slides?
Try the down arrow.
Nope.
Try clicking on the screen. Hit the slide.
Okay. There we go. So some of the species is federally threatened and state endangered. Some habitat requirements include that it needs a high quality wetland like sedge meadow, wet prairie, mesic prairie, marsh, or even bog. It needs full sun. It needs a specific soil mycorrhizae for successful germination. It's pollinated by night flying hawk moths. It has a tuberous root, which you can see in this photo. It does not spread via rhizomes, and a whole population is only maintained by annual seed set. And once we sow a site with seed, it can take 4 to 9 years for a first blooming plant to appear from seed, if that's even going to take. So this is range wide. For the big picture, this species occurs in 8 states for sure, possibly 9, but as you can see of how many populations per state, Indiana, Maine, and Missouri only have 1 population. And in Indiana, that 1 population consists of 1 or 2 blooming plants every year. Virginia possibly has 1 population. Iowa has 5, but the majority of populations occur in Ohio, Wisconsin, Michigan, and Illinois. And I want you to look at this photo. This is a picture of the orchid population at Nachusa grasslands, and you typically will not get this many blooming orchids in 1 photo. So this is a really cool picture. Oop. Well, I don't know how to go back. Let me see. I missed one there. So the recovery plan, there is a recovery plan written for this species. It says that in order to recover and delist the species, we need 22 populations, and these 22 populations have to be highly viable. So on the left here, I've listed the viability criteria that we use. Each of these criteria are ranked from 0 to 3 for a population. I'm not going to read through those, but these are like the highest ranking criteria you could have for a population. Currently, right now, we have 95 populations range wide. I just did a 5-year review. Every 5 years, we review all the populations for their viability. Out of those 95 populations range wide, only 12 were highly viable. Illinois had 5 of those, Michigan had 1, and Wisconsin had 6. And then 29 were moderately viable, 51 were of low viability, and 3 we had insufficient data to determine. So for the 40 or more populations in Illinois, we're fortunate enough to have a great volunteer network with about 70 volunteers. And these are just a couple photos showing that we could get our work done during COVID. This is a group of volunteers at our Wrigley site, which these are Abbott employees. Abbott owns that land. And we're very grateful to the staff at Abbott for taking the species that occurs on their land under their wings. That was not always the case at the sites. So at all the populations, the volunteers take demographic data on the plants. They hand pollinate the plants. They make and put deer cages over many of the plants that they hand pollinate, because a lot of times it takes time to hand pollinate a plant, and then you'll come back in August and the deer will have nipped off the top. And so we make deer cages we try to make deer cages for all the plants that we hand pollinate. And then they also monitor the seed production. If it wasn't for these volunteers, we couldn't get this work done. And you got to remember, when they're doing this work, it's like end of June, beginning of July, it's 100 degrees. It's usually 98% humidity, and they're working in a wetland. So we determine in Illinois our success by how many blooming plants we have. And last year in 2020, we had 2,287 blooming plants out of all our Illinois populations. And that's the highest number of blooming plants for any year in Illinois since we started keeping records, which was in 1991. I mean, we weren't responsible for a lot of that, or weather has a lot to do with that. I have this graph here showing, so on the bottom, this is 1991. These are years all across, and you might not be able to see it behind the people's pictures here. This is the number of blooming orchids, but for 2020, we're way up here. So I mentioned that the orchid is pollinated by a nighttime hawk moth. This is a photo of one. This is Litnaria eremittis, or the hermit sphinx. So the photo actually shows its proboscis unfurled with a simple sewing straight pin used to unfurl it. It's a smaller moth, you can tell by the size of the straight pin. And the yellow packets are actually the pollinia of the eastern prairie fringed orchid attached at the base of it. So it's believed that pollinator numbers for this orchid are low, and that's the reason why our volunteers started hand pollinating these plants before I even came into this program. A few years back, we had a Northwestern student, Claire Elwinger, conduct a range-wide genetic study of this orchid. And one interesting result was that she found that all the populations range-wide are genetically similar. So this is a map showing the 40 populations that she selected across the range for sampling. And she used the results of her genetic work in a genetic computer program that assigned the genetics of this orchid into only 2 genetic clusters, which you see as the yellow or the gray pie charts. And because of that, it suggests that all populations range-wide today show similar genetic structure with high genetic similarity, which is likely due to historically high connectivity between all these populations. So through her study, we have no qualms about moving seed or pollen across state lines more than 50 miles. You know, 50 miles is kind of like the distance that most people feel comfortable with moving pollen or seed around, but I'll explain later, we have done a lot of that across the states. So we're, let me see here. This is a photo of somebody hand pollinating. So we're taking pollen from one plant and hand pollinating another plant. This is what it looks like in the field. It's hard to tell whether this person is taking pollen out or putting pollen in. I wanted to show you this photo. This is a close-up of the orchid flower. These are what's called the anthers, or the pollen sac. We call it pollinia. This is the nectar spur opening where the moth will place its proboscis. And this is the stigmatic surface, this white triangular area. So we take this pollen off. Here's another cool photo of the stigmatic surface. And I'm using these because I do have to teach people how to hand pollinate, not in person with them now. This is what the pollen looks like really close up. This is a pollen sac. We put it on toothpicks and then we just put it in this styrofoam thing and then carry it to another site. This is another really cool photo from Lisa Culp. So here is a toothpick. Here's the pollinia. And when you touch the stigmatic surface right here, if you're in the right spot, now remember, these are magnified. So when you're in the field doing it, they're really a lot smaller. But if you pull that pollinia back, you'll get this string of like pollen, and then you know you've hit the right spot. These are results showing seed set for a plant on the left that was not cross-pollinated, except maybe this seed pod. And I have a yellow raincoat in the back just because it was raining that day. This plant, almost every one of these flowers was hand pollinated, and you can see by the plump seed capsules. Years ago, research was conducted telling volunteers to hand pollinate only half the population and a third of the flowers. But we have results from some sites where they didn't listen to this information, the volunteers at the site, and they hand pollinated way more. And their populations over 5 to 10 years grew dramatically. Like they have last year, 300 blooming plants at the site. So I'm pushing with the volunteers to spend your time hand pollinating as opposed to taking all the demographic data that we need or that we think we need. So I'm going to switch gears now and talk about a problem we had with Maine. So this population is not from Maine. This photo is from an orchid site in Iowa. This was at dawn, just showing how pretty it looks. But the challenge was, or is, in Maine, they have one population owned by TNC. And you can see from these years on the left, they either have one blooming plant or zero blooming plants, 1 or 0, 1. So the thought is this population is going to blink out unless we do anything. So we decided to cross, since they have one blooming plant, the closest population is probably in Ohio. They don't have any pollen from another plant. So we decided to send them pollen and then to also send them seed every year, and to do this for 5 consecutive years and then see what happens. So we're in like, well, the first year was last year. This map, I just want to show you up here in the right-hand corner. This is the Maine population. This is Michigan. This is one of our most northern orchid sites in Michigan. I was trying to get the most northern in latitude to send them pollen and seed. And then we sent them pollen and seed from this site in Michigan, and we sent them pollen and seed from this site in Wisconsin this year. And we're going to do the same for the next 4 years. I talked about teaching people how to hand pollinate virtually. This is what I did. I had to teach the Wisconsin and Michigan folks how to take pollen out of their plants, and then the Maine folks how to hand pollinate. So this is just the container we use to send the pollen. This is glued on the bottom, and it was overnight FedExed. The sad thing is this year, the Maine population did not have any blooming plants, so they couldn't use the pollen this year. Talk about some ongoing research. We know that successful germination of this orchid seed cannot be accomplished unless the seed comes in contact with a soil mycorrhizae or fungus. So we have Dr. Elizabeth Esselman. She's working on determining if the orchid populations across the range have the same or use the same mycorrhizae. So first she collects root samples from young orchids. So this is a photo of a root. She has collected from Illinois, Iowa, and Wisconsin populations. This year she's going to 3 in Michigan and hopefully 2 in Ohio. So she collects the root samples, takes it back to her lab where she isolates and identifies the mycorrhizae. And she has recently furthered her research by she just tried to grow orchid seedlings in a petri dish with the mycorrhizae. And so this photo on the bottom left shows this is a good thing. These, I forget what these are called. That means it's made contact with the mycorrhizae. And then behind this photo, or behind the people here, you can see a little seedling that she had about 5 of these that had like one tiny little leaf, and she outplanted those this past year at Nachusa. So we're going to see if they survive. That's usually where somebody who tries to grow them has a problem getting them to survive out in the field. We have an upcoming manuscript to be published. We've been collecting data from this plant from 1991. And so years ago, Dr. Tim Beller in 2015, he put all the data from 1991 to 2015 from 36 Illinois populations. He used close to 6,000 tagged plants. He put that all in like a statistical analysis and came up with some conclusions. Well, we're adding data from 2012 to 2020 and then going to get this published. But what Dr. Bell found was that most plants only live 1 year, 98% live 5 years or less, only 2% live 6 to 12 years. Survival is highest in relatively large plants, and prescribed burning, of course, increases population survival. So we're kind of excited to see if adding the extra data still creates or gives us the same results. And with that, I'm going to turn it over to Lori Ryan, who's going to talk more about the local orchid populations.
Thank you, Kathy. And I just want to take a moment to say thank you to actually both Kathy Garnes and Kathy Pollock for their wonderful presentations. When I first started working with the McHenry County Conservation District, I think within the first year or two, I met both of these ladies out in the field, and they taught me how to identify the different orchids and actually how to learn to do the hand pollination. And I think it was either 2008 or 2009, Kathy Pollock and I spent some time out in the field in the evening trying to do some moth surveys around one of the populations. So it's definitely been a fun learning experience over the years. But yes, my portion of the presentation is going to be relatively short in comparison, and I'll be talking to you about our local populations and recovery efforts right here in McHenry County, Illinois. So I had a look back in our records and found out that our populations were first discovered on district sites in the early 1980s. And I believe that was through the early phenology program that was kind of spearheaded by Joyce Purbex, where the volunteers and staff were kind of heading out and looking for all the different remnant natural communities on our sites, and definitely searching out for any rare plant populations. And they were successful. The first orchid population was found in 1983 in a sedge meadow. And the next year they found another population at a different site in a remnant dry mesic silt loam prairie. Well, many years went by, and I'm sure many rare plant searches were conducted without any luck. But over 2 decades later, in 2008 and 2011, we finally had some more success, and a few more naturally occurring populations were discovered on our district sites. And in the recent 5-year review that was done by the Fish and Wildlife Service, all of our populations are currently in the low viability or the moderately viable categories. You can go next, Kathy. Thank you. So district staff and volunteers have been monitoring the prairie fringe orchid populations that they have found annually since the early 1980s. And I think what's even more impressive is that we actually have notes in our files about the monitoring counts and all the recovery efforts back in those early days. And so when I looked back in the files, I found out for 1993 was the first documented year of hand pollination, seed collection, and regional seed sharing. And I just wanted to share a few of these dates with you guys today. I know there are a few people in the audience, at least in the chat, that are seem to be familiar volunteers with the program, so they can kind of relate these dates to their experiences on their sites. But in terms of the phenology, it's pretty accurate in my mind compared to the sites that I work on. So July 13th of 1993, the populations were in full bloom, and they went ahead and hand pollinated. Then several weeks later, September 21st of 93, they actually collected 40 mature seed pods. A week later, 10 of those pods were shared with the Nature Conservancy, and a few days later, the district received 10 of those pods from a Kane County site. So this is just really inspiring to me because I had to look it up real quickly, but Kathy can confirm if this is right or not, but the orchid was actually first listed as federally threatened in 1989. So it's kind of neat to see that, you know, orchid work was happening prior to listing here in McHenry County, and then shortly after, you know, the listing, then the Nature Conservancy kind of spearheaded a volunteer network with monitoring, and McHenry County was right on it and active, and our staff and volunteers were already engaged in the recovery of the species. Thank you. And current recovery efforts, since I've been working with the district, we've maintained that annual population monitoring. We've definitely been doing the demographic monitoring consistent with the protocols set out by the Fish and Wildlife Service. So here's just some more pictures to kind of support what Kathy has already showed you already. Little tags in the ground. There's in the top left here, just an emerging Eastern Prairie Fringe orchid, which is kind of fun to see in the spring months when you first get out into the field season. Thank you. Yep, there it is. And then moving down in the picture below that, there I am with a group of summer seasonals and interns. I think it's the highlight of their summer when they're told they're going to go out and monitor prairie fringed orchid. In addition, they're going to learn how to hand pollinate. It's a pretty amazing experience for everybody. But here we are. We measure the height of our blooming plants just up to below the inflorescence. We take the time to count every single flower, every single leaf. We document that. We look for insect damage. If there's been deer browse, we'll make a notation of that. And everything's documented on the monitoring form and then sent into Kathy. And that'll be further sent on to Tim Bill and the others that she was talking about who are running the research projects to kind of, you know, make some sense out of all the demographic data that's been collected throughout the years on our projects. The bottom right picture.
Oh, sorry.
You're okay. I just want to point out, this is pretty incredible. If you can zoom in and look real close on your screen, this orchid was over 1 meter tall. That's one of the tallest Eastern Prairie Fringe orchids that I've ever seen. So I had to have a picture by it. But sometimes they can be, you know, 1 foot tall, and then other times they can be over 1 meter tall. So it's really quite a variation in height for the species. Go ahead. So like Kathy said, we've been conducting hand pollination on our sites. For many years, we followed the protocols, and we probably just said, you know what, we're going to do 3 or 4 flowers per plant. So we'd have our crew of seasonals and volunteers and interns out there, and we would take pollen from one plant. And you can see the plenty on the toothpick here. And then you can go ahead and take it on to the next plant. So we'd have some people just focused on the monitoring, and then we'd have a second wave, a second crew kind of following up behind us to go ahead and do the hand pollination. In recent years, and with the recent research results, we've really been pushing to take this pollen and cross-pollinate between populations in McHenry County. And Kathy will even be great and, you know, ship me some pollen or drop it off some days, and she'll bring pollen from another Chicagoland site, and then we'll have even additional pollen to put out on our sites to augment the diversity. So we can move forward. Kathy showed some really good pictures earlier, but I wanted to show you these as well. So, you know, late September, we'll actually revisit our orchid sites, and we'll look at the production of the seed capsules, and we'll take some additional monitoring notes on those. And as you can see, this is every orchid monitor's dreams. There's maybe 1 or 2 capsules here that are really skinny. Otherwise, the rest of them are nice and full and plump, meaning there's going to be a lot of likely viable orchid seed. Hopefully that'll start some new populations somewhere. The picture on the left was taken, I want to say that's late August, and on the right, then about a month later in later September. So you can kind of see the difference. And once that time comes in late September, we'll coordinate with Kathy and we'll discuss plans for seed collection and sowing, and where seed's going to get, which sites seeds are going to get placed on. And then also, maybe I'll share some seed with her so she can share it with other partners in the Chicagoland, or, even she'll send me seed and I'll put it on some of our sites. Okay, go ahead. So for quite a few years in a row, we're looking at our seed capsule monitoring data. And it really pointed to the possibility that we may have the presence of pollinators on our sites. So here's just a few lines out of our data. And if you look at this, we have the tag number on top. So we'll start up in the top left, and each row would be a blooming plant. So data that's been recorded from our demographic monitoring for one individual blooming plant. So that plant had tag number 5402. It had a height of 93.1 centimeters, a whopping 26 flowers. That's a lot. And you can see here, going all the way down the chart, we're pretty consistent with that 3 to 4 flowers being hand pollinated. Now let's wait our 6 weeks and we'll come back. And if our hand pollination was successful and we did a good job of it, and we didn't have natural pollination, you'd hope to just see 3s and 4s populating that whole column. But what we have here is consistently numbers above 3 and 4. We've got anywhere from 6 and 7 all the way up to, I think, 15 there is our highest number. So we're really excited over a few years of seeing this data that we might have the presence of the natural pollinator, the hermit sphinx, as you can see down there in the bottom left. So finally, after, we can proceed to the next slide. Thanks, Kathy. So finally, after a few years of seeing these trends, we recruited our wildlife ecologist, Cindy Jablonski, and her many trail cameras for all of her wildlife survey work. And we asked if she would help us out by putting some trail cameras on our orchid sites in hopes that we could observe our pollinator. And so we actually only installed 3 trail cameras. This picture was taken after the orchids were in full bloom. So you kind of have to look around the purple circle, but they're actually 2 would be orchids kind of in the immature fruit stage there. There you go. There's one and then one on the other side there. 2 orchids there in that field of vision, but we recruited her to help us out. And unfortunately, it didn't take video, but just anytime there was some slight motion, it would capture a picture. So that's fine. You can go ahead to the next picture. So I think we had those cameras out there for 2 to 3 weeks. And then when we got them back, we started to look at all the photos. And Cindy did a great job at searching through literally hundreds, if not thousands of pictures. And we were very much rewarded when we found a few shots of our hawk moth here. So we definitely got confirmation of our natural pollinator. The time and date stamp is hard to see on my screen. It's being blocked out by the Zoom. But I think this one was July 9th. I want to say it was 72 degrees, if my memory is correct. And it was about 9:30 at night. So like they mentioned before, these are night flying hawk moths. The orchids have a fragrance at night to kind of lure in the hawk moths. And so you can see this one right here. I don't know if it was heading into the flower, if it was actually pollinated, and if it was backing out. But then, yeah, the next slide there, I believe it's the same plant. And this is taken, about 40 minutes later, a little bit after 10 p.m. at night. And here we can see, maybe it's the same hawk moth, maybe a different hawk moth, who knows, but another one being lured into that plant. So this is pretty exciting for us to be able to actually get some imagery of our natural pollinator. So of course I have to ask Kathy, am I done hand pollinating now? No, we got to keep going with the hand pollination and the natural pollination and keep our populations going. So okay, you can proceed. And since this is a Friends of Hackmatack gathering and meeting, I couldn't not share some information, some more recent information about the status of prairie fringe orchid on those sites. But for 3 years in a row, Eastern prairie fringe orchid seed has been sown on one refuge site, kind of in coordination, of course, with the Fish and Wildlife Service. So some of that seed has come from McHenry County sites, some of that seeds come from other regional sites. We've mixed it together. I think in the first year or two, 2017, 2018, our strategy was actually to zip tie the capsules on some branches of some shrubs and let that kind of naturally disperse and fall to the ground. And I believe in the last year, 2019 there, the strategy was actually changed. We actually kind of scratched the seed down into the ground and make sure it was stuck. And if you guys remember earlier in Kathy's presentation, she mentioned that it could take anywhere from 4 to 9 years to actually see blooming plants from seed. So if we do our math this summer, we should actually get a crew of folks together if we can and start searching the property where the seed was sown to see if we can find our blooming plants. And with that, I just want to say on behalf of the district, I look forward to continued partnership with Fish and Wildlife Service, providing any assistance in surveying new land acquisitions for the orchids and sharing seed from our populations and sharing pollen, hopefully once we get some populations established. And definitely a dream and a vision and hope that one day we can establish one of the 22, at least, of the highly viable populations that's needed to kind of recover the species in the National Wildlife Refuge footprint. So with that, I think Kathy and I can take any questions. Kathy Garnis, we can open it up.
Well, thanks, Lori, and thanks, Kathy, and thanks also to Kathleen. There are a number of questions that have been posted in the chat, and maybe I can kind of go through a few of those quickly, and then others can unmute and chime in when we're through with the chat list. A general question, is there any thought to artificial pollination of the calopogon, the grass pink orchids?
Gretel, do you want to speak to that?
Yeah, I know that Floyd Catchpole in Will County is interested in working on the calopogons. So I think that there have not been a lot of opportunities there have been few flowering of the Calopogon oklahomensis. So that's a hard one to cross-pollinate because there aren't that many populations. And yeah, and I'm not sure about collecting that pollen if anyone's done it, but that's something that they're certainly interested in doing.
Thank you, Gretel. And Gretel, by the way, coordinates the Plants of Concern program at the Chicago Botanic Garden and throughout the region. So thank you, Gretel, for your good work. Steve asks if there are any plans to reintroduce orchid species that have been extirpated from our region.
I'll speak to that as well. I'm not sure the answer. I think a more immediate goal is to not have any more be extirpated from the region. So learning about, I think there's been a lot of recent work in figuring out the requirements for germination so we can think about, reestablishing populations that way. And yeah, just saving what we have before trying to reestablish those that have already been extirpated, I think would be the first step.
Very good, thank you. Someone asked if we know what is the host plant for the larva of the hermit sphinx moth. And is that a regular member of our prairie communities where the orchid occurs?
It is bugleweeds, mints, bee balms. But I'll tell you, I've looked at a lot of bee balm out in the field at orchid sites, and I've never once found a caterpillar or an egg. But these hawk moths are strong flyers, It's not like a butterfly that just, kind of lollygags around. At one time, I spent nights out on the prairie trying to catch these moths to identify them. And they fly like bombers, like over the prairie, like they're on a mission. And when they see an orchid, they just come down fast. So my thought was, maybe they don't have to be at the bee balm that's at an orchid site. They could be 5 miles away and come in to feed on this orchid, but that's their host species. I mean, you can Google search the hermit sphinx and find out a lot of information about it if you wanted to, besides that.
Great, thank you, Kathy. How long does the pollen remain viable after it's been harvested?
So I had a lady, so that we have a site in Loda, Illinois, that for some reason our most southern site blooms before that site that's more southern. So I get her pollen from our most southern site, which is in Will County. I drive it to Loda. Her plants are not blooming yet. She's kept that pollen on her kitchen table for a week and then gone out to pollinate plants, and it still produced plump seed capsules. So that we know. I mean, that was just on her kitchen table, but if I know somebody's going to hang on to pollen, I tell them to just put it in a cooler, and open the cooler at night so that it doesn't get moldy or whatever, but then, keep it kind of cool as opposed to hot.
Great. One more. How do you harvest and distribute the seed that you take from these orchids?
You just pluck the seed capsule off. I mean, it comes out like it looks, and we try and collect them before the seed capsule actually splits, because the seeds are like dust. And there's upwards of 4,000 seeds in one capsule. I mean, if we happen to get them later in the season, we just put them in a white envelope, and then whoever's getting them, we just tell them, make sure what you see at the bottom of the envelope gets out in the field, but you just pick them off. I don't know how else to explain it. They come off easily.
Well, that's good to know. I would invite our guests, if anyone wants to unmute and ask a question, you certainly may. I think everyone has the capacity to unmute themselves. And while we're seeing if there are any questions that come up, I want to just reiterate the thanks to our speakers, to Kathy Garness and to Kathy Pollock and Lori Ryan. Wonderful work that you're all doing, and we all benefit from it. So thank you for sharing your knowledge, your expertise, and your stories from the field.
So I gather that once the capsule splits open, the seeds just fall out and you don't have to scrape them out or anything?
No, you don't have to scrape them out, but we've been putting seed at sites for years, and at one of our sites that had lost the orchids in the early 80s, this orchid, this guy that put the seed out kept track of how he did it and where he did it. And he has 2 blooming plants this year from an area where he scraped the ground first and then crushed the seed capsule to make sure the seed makes contact with the soil without digging it in or burying it. And so we've kind of switched the mode that we used to put seed out at a site when we've been telling the volunteers to have it make contact with the soil. In the past, we thought we used to tie seed capsules up with thread or yarn or something to something high in the prairie so that the wind could disperse the seed when the seed capsule split. But what this one volunteer found was when he went back in the spring, a lot of that seed was still in that capsule that was tied up. And so we're like experimenting still after years.
Wow, that's great. Thanks.
We also had a question about what kind of deer fencing you employ.
We make deer cages. They're just circular cones of fencing. I just get it at Home Depot and we actually make them, and it has like a 2x3-inch opening. One of our volunteers thought, because we thought with that big of an opening, a natural pollinator could still get in. One of our volunteers with her research kind of in the field, anecdotal, thinks the pollinator cannot get into the plant anyways. And so she started just taking rolls. And if she has 10 blooming plants, rolls of the deer exclosure to exclude them from like 10 plants at once. So at least the top is open, you know.
Hmm. Hmm.
Well, very good. Thanks again, everybody. I'm going to go ahead and stop the recording.